Hello and welcome. Today on BIOS Frontier Science, we're thrilled to welcome Chris Mason, Associate Professor of Physiology and Biophysics, as well as Computational Genomics at the Weill Cornell Medical College. To help host this episode, I'm joined by my colleague, Drew Yashin. To kick things off, Chris, can you share a brief personal intro with us?
Sure, happy to. I recently was promoted to full professor, so now it's gone from associate to full professor. So am now but I'm just a geneticist through and through. So I'm born and raised in Wisconsin. So I'm just a goofy kid from the Midwest, and have some Midwestern wholesomeness that led me towards a curiosity in genetics. Ever since I was in 8th grade, I've always wanted to be a geneticist, and I'm very fortunate that that is exactly what I get to do. And I was fascinated then as well as now by the fact that we started as a single-celled embryo, and all the instructions for the synthesis of all the cells in our body is present in that very first cell. And when I was a kid, though, we didn't even have the book of life sequence. We didn't have a human genome to look at. We didn't have a template on which we could map functions. But now we have many genomes, not just human, but other species, and start to learn how genetics unravels the biology as we see in the world around us. So went to undergrad in Madison and genetics, did my PhD in genetics at Yale, did a postdoctoral fellowship at the med school, and then also a fellowship at Yale Law School, and been at Cornell since 2009, and have a lab that does synthetic biology, cancer biology, and basically a lot of clinical genetics, and then computational biology to build new algorithms to process large amounts of data. And yeah, that's it in a nutshell. Also spun out a few companies out of the lab. I am a father, a husband, a scientist, runner, poet, sometimes warrior poet if there's a battle. That's it in a nutshell.
Oh, I wish we had a question asking you to do a poet slam, but at the moment, maybe that'll be for next episode. Thank you for that phenomenal overview. Before diving deeper into specific projects, one question we love to ask our guests, and something you started to address. Throughout your career, what would you say has been your North Star? The common thread, if you will, that ties your work together?
It has been the survival of life as we know it in the universe, which sounds a bit preposterously large, but quite literally, I've been thinking about this since I was a kid, and just recently published a book about it, which we'll probably talk about in a little bit. But is ever since I was a kid, there's a few basic facts that I couldn't get out of my head. So the first one is just that the sun's going to engulf the planet Earth in about 5 billion years. It's like something that no matter how amazing we might be, if we have world peace, if we have beautiful technology, and say there's not even any death anymore, but you can't live in the sun, right? So at some point, we have to eventually leave the Earth if we want to survive beyond the life of this sun. It's just a fact of the universe. But so that's something I thought about since I was a kid, which is kind of terrifying. Even though it's far away, it's just this inevitability that was weighing on me ever since I read about how planets form and stellar evolution proceeds. And so with that as a fact, it means that we need to better understand how to survive in space if we need to, by that axiom, leave this planet and go to another one, go to another solar system. So I've always thought, well, genetics is kind of the passion of the science, and the motivating North Star is we need to use the tools of genetics and medicine to be able to understand the body, to understand biology, to build systems that can help us survive. So it's always been, how do we integrate genetics and sort of functions of aerospace medicine and general space biology to enable us to get to the other planets, to survive in the long term. And so that's what led me when I first started my lab. The very first grant I wrote was about doing a kind of like what was an astronaut study, which became the twin study, which was to see what happens to the body in space. Can we learn about it more? Can we have better interventions? How do we, not just physically protect or pharmacologically protect, but at a molecular level, protect our astronauts and the food they will need to survive. So the North Star has always been, I guess, the star that's in the next solar system. I'd say a literal star is what's been the motivating factor.
I think that's an incredibly unique and powerful answer. And you're right, we are excited to dive into all the above. But to do so, I'm going to pass it off to Drew to talk about that genetic basis for human disease and physiology and how we transition and get there to talk about and think about how do we move forward as a species. Drew?
Yeah, thanks so much, Chris. And thank you as well, Chris Mason, for joining us. We're so excited and just super ready to dive into your work. And so kind of just, yeah, just kicking things off here. Your lab develops and deploys new biochemical and computational methods in functional genomics to elucidate the genetic basis of human disease and physiology, with a particular emphasis in oncology and on the elements that orchestrate the development of the human brain. Just starting off here, to provide a bit of context for our audience, Chris, can you share a brief history of functional genomics?
Yeah, good question. So functional genomics is, for people that don't live and breathe it every day, it's, those two phrases are key in the sense that The genomics is the genome, so the the template of DNA that is just, I was just saying, really creates the synthesis of all the types of cells in your body, makes your muscles, your neurons, makes your enzymes, makes your skin and your hair. That makes you as an organism. It's true for all organisms. So the genomics is the DNA and all of the layers of the DNA's regulation, or what some people now call multi-omics. Like, I mean, there's peptides and lipids, and there's RNA and DNA. You could say omics in general, but genomics is a general term, is the DNA and all the things that regulate it and how it's active. And the functional part kind of is an adjective, just really points to what is it doing. Like, we have 3 billion letters of genetic code in almost every cell in our body, and we only really have a good sense of what a few million of them, maybe hundreds of millions of each of those letters does, or likely does, or what it changes in response to a poison or food, or when you go take exercise. So the functional genomics part is to tease out, okay, we have this extraordinary genome that is part of our heritage, shared genetic heritage. What does each base do? Do we need all 3 billion bases? Could you get rid of half of them and still be fine? 10% of them, 1%? Now, we do know, for example, if you have Down syndrome, you have a whole other copy of the chromosome, another several, but over 100 million bases just like added to the 3.1 billion bases. And that's enough to cause disease and have severe problems, right? So you can sometimes have too much, too little DNA, but at the end of the day, the functional genomics is we have these letters of genetic code. What are they doing? Why are they there? How do we understand them in health as well as in disease?
Yeah, that's kind of amazing. Kind of expanding on that slightly, really building out that research. What is a metagenome, and how does it link back to functional genomics?
Yeah, great. So the metagenome is kind of, the name implies, it's like, broadly includes all genomes. It's like, or I guess now Meta is the name of a company for what used to be Facebook. The meta is like everything. It's this whole universe. And metagenome is all of the genomes that are kind of in a given sample. So not just the human genome, or say, if you have a bacteria on your hands, the bacterial genome. The metagenome is all the DNA from any sample. So originally it got started in environmental sampling. So say you go to a lake, go to a river, go to the ocean, and you sequence all the DNA that's there. It's a metagenome because you're going to get plants, animals, bacteria, viruses, fungi. You get everything in there, and you have to analyze it not just through the lens of one organism, but through all of them, or the metagenome. And that links back to the functional genomics, because our physiology is not just human cells, and it's not just human biology. As anyone who's had diarrhea can attest, a microbe can wreak havoc on you quite distinctly, or anyone who's gotten COVID, right? In the severe case of COVID, we know that other organisms can severely influence our health and also disease. But the metagenome is changing how genes are regulated in you. It's actually changing your biology. Basically, they link together because you want to view biology as an ecosystem. Because so if you treat a patient, you don't just treat the human cells, you treat all the cells in the body, some of which could be bacteria. So you take antibiotics to kill something that's giving you an infection, or an antiviral to stop a viral infection, right? So if you view health and disease through the lens of the metagenome, that gives you sort of a functional metagenomics, which is, what is any piece of DNA doing at any point in my life? And I want to know that, and then treat it, or in some way use it. Yeah, definitely.
Yeah, 100%. And now kind of bring all these two veins together. To bring the field of functional genomics forward, your lab has pioneered a number of techniques in next generation sequencing and released 12 open source software packages for everything from DNA and RNA modifications, genome, epigenome engineering, to tumor evolution and genome evolution. Crazy, by the way. It's amazing. Among these techniques is the MeRips sec, right? Which uses antibody specific for M6A sites, and is coupled with the computational pipeline for MeRipper to study M6A significance in cancer and viral infections. Just kicking off here, can you share more about the significance of M6A and the early outcomes of the studies?
Happy to. So really, there's this, a lot of people hear of genetics and epigenetics. They'll think, okay, DNA, of course, is genetic code, but epigenetics is the layer on top of DNA that changes when and how DNA gets used. For example, the DNA in your neurons in your brain is the same DNA that makes your muscle cells and your heart cells, but it's just what's turned on or what's turned off is what's different. It's the same DNA, it's just controlling what gets used. And the same thing is true with RNA. We have all these RNAs, which is the active form of a gene where it gets transcribed and becomes RNA. So it's kind of these other molecules that are, really very similar to DNA, just adding one oxygen. But the same principle applies there. There's RNA molecules, are these very long molecules that can fold on each other. They have their different functions. They're out there performing these, real discrete tasks for cells in the body. But on top of them is also this epitranscriptomic layer. So the transcriptome is all the RNA in a cell or a sample, and the epitranscriptome is just like the epigenome to the genome. It's the regulatory layer on top of the transcriptome. So in M6A was something we discovered with Simon Jeffrey's lab in 2012, was something that was thought to be pretty rare on RNAs, like appearing just a handful of times. But we published a paper and submitted some patents back in 2012 showing that not only is it in more than one or two places, it's actually pervasive across almost every gene in the human genome, that when it's transcribed and becomes RNA, a lot of them have these slight chemical tweaks. And at the time, we were pretty intrigued because it looked like it controlled splicing, how RNAs get combined, controlled stability and movement of the RNA. And so it really added these other kind of like layers of control onto the RNA. Just like you think of DNA and packaging and modifications for the genome, RNAs have their own control levers. And so we published this method and patented that technique to basically make it easier to find all these modifications and better understand it. But the field is still developing because it's really only about 10 years old, and we're just started that back in 2012.
Yeah, like while you're developing, as we're on this topic, by the way, are there any other NGS techniques or software packages that your lab has developed that you just like to highlight real quick?
Yeah, one of my favorites is actually the one most cited is called Methyl-Kit, which is a way to analyze epigenetics data, and it's basically just an R package with some tutorials that helps you process a lot of large data sets in epigenetics. And so that's actually one of the things I've been very proud from the lab is all the tools are open source, open to anyone to use, and they often come with tutorials. So if you're like, I don't know how the hell to do this, and you just think I'm not a computational biologist or a bioinformatics guy, but try to make it easy for anybody to learn the tools and the techniques that we've developed over the years.
That's fascinating. I think at this point in the YouTube video, I'll link up to a source on your website where people can find this. We love accessibility. It's a big thing on BIOS. Yep. So thanks again for your work. Continuing for a moment on your oncology work, your lab has closely worked with researchers from New York Presbyterian, MSK, NYU Medical School to define genetic subclassifications of cancer patients and better develop targeted treatment regimens. Can you tell us more about your work in this space and kind of get things going here?
Yep, definitely. So this is an extraordinary time in medicine that we have, customized therapies for many diseases that used to be completely lethal. And among them are things like CAR T, for example, which is a chimeric antigen receptor T cell. It's basically an engineered T cell that you can pull out of a patient's body, reprogram it, essentially add new features, then grow it up and infuse it back in, and really open up a new therapeutic direction for a patient who maybe didn't have any before. And so it really is this extraordinary area. We've published new ways to design T cells that can basically target cancers that previously you couldn't touch before. The other thing that's just in precision medicine is, you think about if you have a cancer anywhere in your body, you might think, oh, it's a lung cancer or it's a liver cancer or prostate cancer. We often think of cancers to where you found the cancer, which is important. But what's become more apparent in the past 5 years is it matters, but it almost doesn't matter as much in many cases because you want to know the mutations that are in the cancer, not necessarily where it came from, because that guides you towards what you can treat it. So it's different, cancers anywhere in your body have different mutations, but many of them are exquisitely targetable. So you can say I have this one drug that targets any cancer with this mutation and can lead to some cases a complete cure or at least long-term remission. So really, extraordinary potential, I think, for what we already learned, but there's even a lot more to come in terms of can we find other mutations and target other cancers with customized immune systems that we're just building for the first time.
Yeah, we definitely love your work in CAR T. I think it's been phenomenal actually seeing the impact, especially in precision oncology, and just continuing down that vein. This is something we just love continuing on with our guests, especially with a precision medicine focus. Speaking of what's to come, in your view, what's really coming next, and what are you most excited by within precision oncology itself?
Yeah, the customized immune systems are probably my favorite, I think, because we now know enough about what changes in immune cells. But there are other areas to think about, not just treating on oncology. You could also, even replace entire organs. There's a effort to actually do xenotransplantation, so you modify organs from pigs and use those. And even organoid modeling, for example, you take out someone's cells, make a mini brain or a mini liver, and try a lot of drugs on that avatar and see what works, and then use that on the patient. So a lot of the precision oncology and precision medicine is growing little versions of yourself and then seeing what works there. And it's not perfect, And it gets a little creepy if you're like, oh, that's a piece of myself growing over there, and there's a little piece of my brain, a little piece of my heart, my liver, what are called these sort of organ systems on a chip, which are still imperfect, but they're getting better. And it does open up these doors, though, to a really broad view of your body, kind of disassociated, but then tested, learned from, and then brought back to your body for deployment of any therapies, which is pretty cool.
No, it's super exciting time. I love the overview. Now, kind of shifting our focus away from oncology now to neurology. Your lab at the same time also generates detailed and cell-specific molecular maps of genetic, epigenetic, transcriptomal, and translational activity, creating a draft of a molecular recipe for the creation of the brain and part of a non-human primate reference transcriptome, what is called just NHPRT, the NHPRT project. When coupled with transcriptome and the epitranscriptome mapping, this baseline serves as a starting point for understanding neurogenetic disorders. From your perspective, we love to learn more about the launch of NHPRT and insights that really this is just generated today.
Yeah, so it is. It started actually, as many things do, with some people complaining at a bar, and someone saying, "Well, there's, I was at this, it was a genetics meeting, and someone's saying, we have, because at the time, we talk a lot about sequencing already, that sequencing is ubiquitous and it's now cheap. And at the time, people were saying, we use all these rhesus macaques for vaccine testing, for animal models, for diseases, but we didn't know how many genes they had or where they were, or what were active. It's the same question of functional genomics, but just now in primates. And so the NHPTR was just an effort started at the bar at a conference saying, sequencing RNA is not that hard now. We can do it pretty well and pretty cheaply. Why don't we just do it for a bunch of these primates? So it was the same quest of functional genomics just applied to another species, in this case, primates, to say, what's active in their brain, what's active in their tissues. This is something that we can actually quite imminently solve with just sequencing the RNA from different tissues. So it's become the reference map essentially for the field and for many species, actually over a dozen different species of primates that we analyzed and sequenced, and that have contributed towards that resource.
Yeah, and as you bring this new approach forward, what do you believe the greatest challenges will be?
I think some of it is the sparsity of functions. Sometimes you only need one gene at a very certain time in a very specific place. And so even if you take 100 adult primates or chimps, or even 100 humans, and you sequence all their RNA, you think, okay, now I've got a good functional map of what genes are active and what they're doing in different tissues. And there's a project in humans for this called GTEx. The genome and transcriptome expression analysis, basically GTEx, is something that just in humans is doing this, this very work. So it is, but the problem though is the genotype tissue expression kind of maps. If you only have something that's turned on when you're a fetus, then you can only learn about it if you have fetal samples. A good example is fetal hemoglobin, for example. For all humans, you have a different kind of hemoglobin that carries oxygen in your blood when you're a fetus versus when you're a kid or an adult. And what's extraordinary is that but you wouldn't know that unless you're actually grabbing fetal samples and sequencing them, which can be hard to do for obvious sort of ethical and sort of just clinical reasons. But that's what's hard, is you might always miss something because it's only in a certain cell or in a certain time of development. But when you do map them, though, you can use them for really extraordinary things. So one of the best clinical trials I've seen in the past year is reactivating fetal hemoglobin in patients actually that have beta thalassemia or sickle cell, because they have a problem with their adult hemoglobin. So the idea for the trial is, well, if they still have the gene for fetal hemoglobin inside them, why don't we just turn it back on and see if that could fix their disease? And indeed it has. So it's really extraordinary to think, there's genes inside me, inside all of us, that were on when you were a fetus or other times in development, that can actually be turned back on to cure a disease. Really kind of the unprecedented time in which we live in that regard.
Love the connection. It seems like almost some of the simplest answers always kind of come back to creating mega solutions, and that's a phenomenal study. I love that. It kind of just continuing this on as well. In addition to your work in NHPRT, your lab is also assessing epigenetic and genetic risk factors for neural tube defects, just NTDs. What are NTDs, and how will understanding their development help us overcome a lot of these defects?
I mean, I did a lot of this work back when I was at Yale, which was looking in families that are inheriting disease, and where they're all seem to, all the children seem to be getting it from, one aunt, or you can see in these big pedigrees, clear inheritance of diseases that would lead to brain malformations or neural tube defects, really severe neurological problems. And this was only possible to map once we had a reference genome that was completed, that we could say, we think we know it's this gene, and then find it and track it down. So it's really the clinical and translational aspect of some of the work in our lab, is to say, okay, well, people are getting disease and it looks like it's being inherited in this very clear Mendelian fashion, meaning it looks like it's clearly heritable. And then you get the DNA, you get a cheek swab or a blood sample, and you kind of track down exactly where it's coming from. And we do this also not just for brain defects, but also inherited cancer. Basically, patients who have a high risk of hereditary cancers, means they're like BRCA1 is the most common known one that Angelina Jolie had, where she had a prophylactic mastectomy to get rid of her breast, just in anticipation she might get cancer, just to play it safe. And so other people also have other risk genes that clearly are driving their cancer. And so we're trying to find them in those patients as well.
That's fantastic. Just building off those two approaches, and really more just taking a step back. As we think about therapeutic applications of oncology and neurology more broadly, really just love to start basic, like what's coming next? Have we reached a point where we can begin to repair, re-engineer, or fortify these genetic networks within human cells? What's the frontier, as we've discussed everything from future of our sun and our solar system? Like, would love to just learn more.
Yeah, that's the long-term dream, is that, can we have all the tools of functional genomics in any species, understand the genomes, and say, okay, well, I think I know what the map is. I would like to then rebuild the map of life itself. And I think that is unquestionably the best test of understanding. I mean, the best example would be like, if you have a chair or you have a computer in your house, can you disassemble the whole thing and then put it back together? That's hard enough, but can you disassemble it and then also add a bunch of other components and then put it back together and still have it work? That's actually very, very hard, and not surprisingly, because you're putting things together that have never been together before. And so they may have worked functionally in their own way in different genomes in different places in the world or the universe, but building them together is both a creative leap and challenge, but also, I think, the best test of, do you know how those functions work independently and jointly? And so I think what you're beginning to do, though, is, like I mentioned, with beta thalassemia and the trial that's targeting it. So basically, it targets something that's called BCL11A, which is an enhancer that just keeps fetal hemoglobin turned off. Anyway, like once we start to find these kind of, they're like light switches in the genome, these things that control very exquisitely what's happening in your cells, we can start to move them in pieces or chunks that actually can change control of other genes. Or one of my favorite examples from the lab that we're using right now is we take genes from the tardigrade, which is this cute little water bear that looks like it's microscopic, but looks like a bear, can survive in the vacuum of space. It has unique properties that let it repair DNA inside of itself. So we've taken that gene from that organism, put it into human cells, and stably integrated them. And now they can resist 80% more DNA damage than they could before. So basically, you can take evolutionary lessons from one creature, like a tardigrade, put it into a human cell, and have it start to have these hybrid functions that really open up a lot of new creative directions for really synthetic biology and genetics, but also for where we can survive. Like, we may have to get that creative just to survive in the vacuum of space, or to survive on Mars or and planets farther away.
No, 100%. That is so fantastic. And keeping things future focused here, I want to pass it off to my colleague, Chris, really to speak on our second topic here and just get as much as we can done through functional genomics consortia and the 10 phase and 500-year plan and more. Chris?
Thank you, Drew and Chris. Building on your work with the NHPRT, your lab is focused not only on the development of techniques and technologies, but also on the establishment of consortia that baseline their application and enhance our understanding of genomics, epigenomics, and metagenomics for a wide variety of applications. So as part of these efforts, first to highlight your lab's work with NIST and the FDA on projects such as SeqC2, IMMSA, and the Epigenomics QC group. You're defining international standards that ensure clinical quality genomic measurements and editing. Can you tell us more about these projects, even just at a high level, and talk a little bit about how they're influencing outcomes of precision medicine?
Happy to. So some of them are extremely dorky projects. They're just, I want to take the same sample and run it in 5 different labs and get 5 similar answers, or ideally the same answer, which you can. And unfortunately, the answers a lot of times you don't. I think, there's always a headline now and again about I did a 23andMe test, or I did this different test and I got different answers for risk for heart disease, or I took my poop sample and I sent it to 2 different labs and I got 2 completely different answers. And this is disheartening because you would hope that science is built on a bedrock of empiricism and reproducibility, or at least it's supposed to be. But we know that there are many reasons why there's challenges. It could be physical heterogeneity, it could be processing, it could be computational methods. There's no shortage of reasons why things can be a little bit different when you extract, process, analyze, or in some way process them. But we've worked with the FDA, with NIST, all these standards groups and clinical groups to make sure that we get as close to perfect as we can. It's kind of the more perfect union of the political view, but applying that to molecular biology. Can we get, probably never going to be perfect because we don't live in some platonic form world of perfection, but can we just get as good as possible? But more importantly, know the areas where we can measure very, very well, reproducibly at 99.99999% accuracy, and we're good. But there's other areas, for example, of the human genome. Sorry, I'm by the hospital. You can hear the siren here. But we know that some areas they're more complex. They have repetitive DNA structures that are hard to see, or that just mismap, or just have challenges. So even just well mapping the areas that are hard to see or more difficult to see is of great utility for the field, for what we run here at the hospital, clinical samples, or what you do in research as well. And so these consortia, or in some cases, some of those dorky people you can imagine. There's actually something in the U.S. called the National Institute of Standards and Technology. There's rooms full of people who just think about standards, like that's it. And you think, oh, that sounds really boring, until you want to measure something. And you're like, oh, I hope that someone knows exactly how to measure this, whether it's, a yard for building a house or a mutation in your genome. And you're like, oh, I need those standards, or else I will have a craptastic time at whatever thing you want to do is. And actually, that's it was in the Constitution to start to establish some of these methods in the United States. Partly because the U.S., the founding fathers, really wanted it to be different from other countries where there were no standards. So it's actually a great heritage of the American scientific system.
No, it's a phenomenal link back to history. I wasn't aware of that at all, and it's really interesting to think about the influence that just establishing standards can have, especially as we think about some of the larger scale mapping projects you talked about. And so as we pivot slightly to dive deep into one of those that you've led, in 2013, you launched PathoMap to create the first genetic map of a city, going back to Drew's early question on metagenomics, and to study New York City's microbiome. This in turn led to the establishment of the International Metasub Consortium, which was launched in June 2015, to build a DNA map of mass transit systems, which seems especially timely to discuss given the recent pandemic. That I'm just going to skip that. Given the impact that epidemiology studies can have. Can you tell us more about these initiatives, their launch, and maybe where the Metasub Consortium is today?
Yeah, which I just landed last night from our annual meeting of the Metasub collaborators, where we had talks from Scott Gottlieb, the former commissioner of the FDA, director of the National Wastewater Surveillance System at the CDC. We had academic, nonprofit, for-profit collaborators all come together once a year. And what's great is, it started as a merry band of swabbers who love to go out and swab things that were mostly self-selected because they'd seen our paper from 2015 where we did this first for New York City. And even that project itself, to get to the genesis of it, was as a young father, when my daughter was about 2 years old, she was out riding on the New York City subway. And as young children do, they explore the world with their mouths, right? There's also, I feel like, teenagers also do this. Young children and teenagers, just kind of 2 times in your life where you explore the world with your mouth. But she licked the subway pole, and just for a second, but it was a moment of pure terror as a parent, like, OMG, what just happened? That has been a microbial exchange. I want to know what it is. What has happened? So you went to go look in the literature. Like, someone must have studied this. There must be papers or analysis of public transit system, microbiomes and viruses and what's there. But there's almost nothing. So I thought, well, let's find out what's there. So I had a bunch of summer students in 2013, and they came and said, Dr. Mason, I'm very excited to be in your lab. I want to study cancer genetics and synthetic biology and genome regulation. And I actually looked at them and I said, thanks for coming to the lab. I'm excited to have you here for the summer. I said, but all those diseases and human problems, they're not going anywhere. We've got like decades and decades ahead of us. But I have a unique idea for this summer. What if we all swab every single subway station in New York City in triplicate sequence it and make a genetic map of the city because it's never been done before. I think it'd be kind of fun. And they were super enthusiastic, jumped right in, built an app, got a crowdfunding campaign going, got all the protocols ironed out, and got started. So it became this really, shooting from the hip, exciting idea for a project that then has now become this international consortium, which, to your point before, was a really peculiar project and a bit out there until the pandemic hit. And then we had a great infrastructure for people with extraction, swabbing, and sequencing and analytical protocols for new viruses and bacteria. So it became very, very relevant, and we've got additional funding from the NIH and from WorldQuant and other and Gates Foundation. So we got a lot of additional funding in the past few years to help with the pandemic.
So as we think about these collaborations and combating that next pandemic, how can we best support and develop consortia like this to keep moving forward and prevent, maybe not prevent, but at least slow the spread or stop something from happening in the future? Is there a way to be more informed, let me say instead?
Great question. So this is actually the big question of public health right now today. And actually, it's as old as John Snow when he collected, data about cholera and the outbreak in London in the 1800s. We can see changes around the world or around a city, and how do we use that data to intercede and prevent a pandemic or prevent infections, to improve public health, to stop people from dying, quite frankly, right? So can we? And it was hard to do because at some point you need to. What John Snow did, for example, it was a public well that people were getting cholera. He took the handle off the well, right? So he said no one can get water. It made it impossible to get water from that place anymore, which is, it's hard today to make that kind of public health intervention that says, I will prevent you from getting the disease that is around you, or at least that directly. But we do still, of course, have vaccines, right? So the rapid deployment of mRNA vaccines is among the most successful stories of medicine that humanity's ever written, and we can now do it much faster. So the sequencing and swabbing and analytics quickly tells us what's mutating, where the virus is, what are the different strains of viruses, and we've already had a booster that has a multivalent vaccine that's already been deployed, and I think we'll see more of that. But the hardest thing is just, we need to also have ways we can monitor how people's immune systems are working. Are they reached immunosenescence, or are they still fully ready to combat a virus that they encounter? We have imperfect ways to measure that today, and so that's getting better. But the analytics help us see where it's moving, how it's mutating, develop the vaccines that get updated in annual or even more frequent basis. And then, what's interesting is I just had a great call last week of, we all hear about CRISPR and editing, and basically CRISPR is a bacterial immune system that destroys viruses. There's even some clinical trials being planned now where we could basically build CRISPR libraries that you infuse into patients and directly attack the virus. But you could actually imagine someone coming to you as a new patient, sequencing it in less than 24 hours, exactly what strain they have, just custom designed guide RNAs against that virus, and have a perfectly tailored antiviral for one patient every time they come in. Now, generally, you'd probably want to go towards the predominant strain because that'll work fine for most cases. But with all the sequencing and analytics and vaccine and targeting technologies, we can really have a brave new world of viral defense and protection.
And I think that world is only continuing to improve. So I'm excited to, with you, hopefully see what's coming next. But as we think about actually this transition and collection of information, I would actually be curious your thoughts, and this is going a little bit off the cuff, into decentralization. There's so much talk about Web3 nowadays and how it potentially links into larger scale public health. I'd just be curious your thoughts on its potential applications here.
I think so. Web3 has a lot of buzz, and it's got its haters and its lovers. It's a bit like the latest dress on in Hollywood. I don't know. It's like some people love it, some people hate it. I think it remains to be seen because it does have a potential to integrate more with people, and I think be useful. But I think it's just too early to tell how useful it'll be, because it's like any new paradigm where there's not as much of an ecosystem, not as many tools, not as many people engage with it yet. So, I think I'm always open to new ideas, and if something looks like it can either engage people more or transmit information better, or, in any way improve how we deploy medicine or track response to it, I'm all in favor of it. It just has to be, it's like anything, you have to get a mixture of the addictiveness of TikTok with the functionality of, vaccine development or something, which hopefully we'll get there.
At some point, that would be great. And I agree with you. I think the infrastructure certainly needs a significant amount of development, but by the same token, always curious to hear from the experts in a space. So transitioning slightly from public health to precision health. More recently, in 2017, you helped found the WorldQuant Initiative for Quantitative Prediction as co-director. And the WQI brings together financial and medical experts in collaboration to analyze genomic data to both identify patients' patterns and trends that might predict patient future risk of developing disease, as well as their potential outcomes. Can you tell us more about the WorldQuant Initiative and its goals for precision medicine?
Yes, so this is bringing big data into small cells. The idea that you want to have as much data about yourself, the world, what's being deployed globally, and build these models, kind of like the PathoMap and the MetaSub models, and also the precision medicine models, all the data from any virus you've seen anywhere. You want to basically bring that to bear into what is your predictive model for medicine. So we often think about precision medicine. So if you're sick, make sure you get the right therapy or the right dose for the right patient. And we think of, I think, actually preventative medicine, and you want to try to avoid it if you can, right? But I also want to think about, predictive medicine as a third branch of medicine that's, it's kind of really supported by this initiative, is can we know that you're on the path towards a disease, but, get preventive medicine, you have a good prediction of what's coming in a year or 5 years or 10 years, and nudge the trajectory, much like if it's an asteroid's coming to Earth and we think, oh, crap, this something's going to destroy us. What if we nudge it early enough with just a bit of a nudge? It might actually be enough to completely avoid a disease. And so one of the best examples of this is something called clonal hematopoiesis, where we look to see mutations in the blood that presage cancer, sometimes by 5 or 10 years in advance, that at least you want to have, more continued and more frequent monitoring, if not even intervention down the road. But that's one direction. And then the other thing is more on the predictive changes for viruses, how they're mutating and evolving and changing. And use that as a way to guide therapies there. Actually, the most unique thing in the entire initiative is we have these quants, people that are quantitative hedge funds that are just doing programming and machine learning on financial data, who come here to play with genetics and biomedical data, and vice versa. We have, dorky grad students and postdocs who spend all their days staring at genetics data and say, okay, I'll spend a summer at a hedge fund and have this very unique exchange of ideas and expertise between the two divisions. And actually, Igor and I, who's the director, we have a book coming out next year called The Age of Prediction, which is a lot about those intersections and commonalities between the fields.
I'm incredibly excited to read the book when it's ready. But in the meantime, would love to learn a little bit more maybe about the elements that support this shift from precision to predictive medicine. Are you primarily looking at things like family history? Are you looking at individual genetics? Are you looking at simultaneously linking in the financial records and information to understand not only where the most impactful health outcomes could be, but financial outcomes given the burdens on the healthcare system today? I'm assuming it's some combination of the three, but would love your thoughts.
Yeah, it is all of them, and even things like social media data or other data that's on dashboards or online, even Google search data. So we try to leverage, for a lot of machine learning applications, it doesn't matter so much why the signal is the way it is or where it's coming from, but just is it predictive and is it a reliable indicator? So some of these same methods are helping us to diagnose infections that are not culturable. I mean, you can't grow them up, but you can sequence bits of them and using some things called there's something that's actually in finance called the Gini coefficient, which is inequality in societies. Do the rich have more than the poor? Well, they usually do, but how much more, right? What's that degree of a difference called the Gini coefficient of inequality? And that's also something we use the exact same formula to look at when you have a piece of a bacteria. Do you get evenness of the coverage of the genome, which indicates you probably captured most of it, or is it very uneven, meaning it's probably not as accurate? So we use literally the same code in finance as well as in genomics to try and track this to get a better accurate read of what's present in a sample. But for the predictive medicine parts, a lot of it is more continued surveillance of the gut microbiome, the genome, the epigenome, the epitranscriptome, all the ohms we talked about. We monitor them more routinely and continually to look if there's any drift towards a disease-like state, and then use that as a better way to hopefully avoid it. So if you can see if it's closer. So a lot of it is mapping and monitoring every molecule we can get our hands on, which we do like for astronauts, for example, and using that as to build the predictive models.
Okay, so in that case, then it sounds like access for these diagnostics to specific individuals is going to be the primary cause that would lead to a tipping point to bring us into this sort of predictive analytics.
I think it's a great, yeah, access to it, and also the willingness for the patients to really deposit samples. We actually ran a trial here during the pandemic of people give blood, stool, urine, saliva every 3 to 4 weeks. just to see all the hospital workers and people working on campus, who's getting a disease, who's at high risk or low risk, what was their immune system, what was their stress, even all things like that, like a survey of just, are you working 80 hours this week or 150? Like, do you feel stressed? It was like these really, interesting surveys in the middle of the pandemic. But, studies like that really help you map out long-term, how are people responding to life and stress and disease. And also the Framingham Heart Study is a very famous example where it started in, really almost 70 years ago now, was just to follow people long-term and say, okay, who's at, what do we think, high risk for heart disease or low risk? And how does that map out over multiple generations? And so we're, trying to do this actually for astronauts, a multi-generational clinical trial of everyone who goes to space and doing all this rich molecular profiling to predict where they're at on a long-term disease risk.
That's a perfect transition. To pivot once again, we've gone at this point from public health to precision medicine to predictive medicine. Let's talk about what we might call extraterrestrial medicine and talk more about your work with NASA to link metagenomics to the design of life for environments such as space. So recently you wrote a book on this topic and you're an author behind the 10-phase 500-year plan. Can you tell us more about this?
Yes, so I wrote this as a blog post for the lab in 2011, was for the International Genetically Engineered Machine Competition, and we were making radio-resistant microbes, and the idea was so they could survive in space. But then someone said, what's the point of all this? Do we have any like blurb of why we should do all this? And so I said, well, yeah, I think this is actually part of a broader view of science and medicine and what we should do and why we should do it. So I wrote this 10-phase 500-year plan for the lab, which I'll be dead for most of it, but the vast majority of it actually. But I think it's emblematic of something that's extraordinary that humans can do, and we don't even think twice about it. You can lay out a plan that is for decades or centuries, and of course it'll change. No plan stays exactly the same, especially the farther it goes out. But the fact that you can do so is a quintessential human trait. And so I talked about the value of humans with an awareness of extinction, with the ability to make these long-term plans. And that means that we're really the only ones in the universe that understand extinction and can actually prevent it. Of course, we can cause it too, and we've done that, unfortunately, with things like the thylacine and other species, the dodo. But we can resurrect species. We can even bring them back. We can actually prevent extinction. We are basically the only organisms in the entire universe that we know of that could do anything about extinction and actually get to the other star someday, like what inspired me when I was a kid. And so now I just have a bit of more of a moral and technical framework behind it, is that we have a duty to do this because we're the only ones that actually can do it, and that we are the only ones that can serve as the guardians of life because of that. So I think this is, I think that life is unusual and life is precious. There are some people who think that, well, if all life in the universe died, who cares? There are some nihilists, and it can't help them as much. But life is about the only other thing in the universe that resists entropy other than gravity at some degree. But it's a unique force in the universe, and I think worth preserving. And of course, we have music and poetry and all sorts of other wonderful things about life, and this complexity and this beauty of life and all sorts of, I think, wonder, which has its own value. But I think the 10-phase plan is the steps by which we understand biology, monitor astronauts, get to other planets in our solar system, and in a nutshell, by the end of 500 years, have all the technology we need that we learned from extremophiles and engineering to be able to put people on a generation ship, which means multiple generations live and die in the same spacecraft on their way towards another planet, in another solar system, which I laid out in the book, are the ones that we could get to and possibly survive today. So it was a fun book to write.
It seems like a fun book to read. Diving deeper a little bit into the actual science behind it, or as you mentioned earlier, you began your career with space, at least in part, through the digital twin study. Can you tell us more about the study and its outcomes for those who might be unfamiliar?
Yes, so it's called the NASA twin study, where there was a twin going up in space, an identical twin. Scott Kelly was going up, and his twin brother, Mark Kelly, was staying on Earth. And he also actually is now Senator Kelly. So Mark Kelly is now Senator of Arizona. So we have the beginning of the Senate Genome Project. But the idea was he's going in space for a full year. We should monitor him and see what happens to him and compare it to his twin. There's also a cosmonaut going up for a year, although we didn't get as much collaboration with the Russians for that study. But the idea is it's a really long mission. There's only been a handful of missions longer than a year. Most of them are 3 months or 6 months. And we started thinking about going to Mars and back. That would probably be about an 8 or 9 month mission. Plus, you got to get back. So we want to start to do more modeling of these really, really long spaceflight, exploration class missions to understand what happens to the body. And so I was one of the PIs for the study where we did genetics, epigenetics, proteomics, microbiome, all the omics of the twins. And we now do this for SpaceX and Axiom and some other spaceflight providers as well. So we want to know what happens to the body in space, and then can we learn from that to have countermeasures and get them ready for Martian and lunar missions.
As we think about the transition now, and we talk about working with SpaceX, working with missions today, how has research into these molecular foundations of, I don't want to necessarily call them genetic defense, maybe genetic engineering, for enabling long-term human spaceflight progressed? What, in your view, is sort of the state of the art and what's coming next? Are we just in the stage of monitoring, or are we beginning to understand which elements of the genome we should really be highlighting?
Yeah, Greg, I think we're still in the early stages of monitoring. We probably still have another good decade of monitoring. Now, keep in mind, we just got the first draft of the human genome in 2001, the first real complete draft in 2003, and the first truly full genome with all the pieces filled in just last year. So actually, there's been these other areas of the genome I've mentioned that are hard to read through. We just finished that last year. So we're still in the early stages of this mapping, of this monitoring, but we already have some countermeasures that we know can prevent the radiation damage, either from tardigrades, like I was talking about before, or from other drugs that target specific genes that seem to really stop the damage. So, you know, we do have some good candidates, and there's an ongoing quest to find better ones, you know, all the time. But we have what we already know now are some pretty good methods through which we can prevent the radiation damage, and you know, it's just begun. So I think after another 5 or 10 years, we will keep narrowing down the targets that are consistent responses to 3 months, 6 months, or 1 year in spaceflight, because right now we don't have that many missions to guess at that yet. And look at the things that are more transient, things that seem like they change a lot in flight. For example, like cortisol, you're under stress, it spikes up, then goes back down, but then it's generally fine again, right? So there's some things they're just a normal response to stress that the body actually, you know, requires, and we definitely don't want to stop them from having their response. And so I think we'll be able to tease those out over the next 5 years, I'd say, and do more targeting of them.
Excited to read more as those insights are generated. For now, though, keeping on this topic of early stage engagement, I'm going to pass it back to Drew for a quick final topic on academic entrepreneurship.
Yeah, thanks so much, Chris. And Chris, building on academic entrepreneurship here and building on our discussion overall, we'd love to learn more about the companies you founded to translate your lab's research in the clinic for patients. Thus far, you founded 4 biotech companies, including Longevity Health, Biodia, UrsaBio, Nurture Genomics, which launched this year. Congrats again. Could you give us an intro to Biodia and share more about its launch?
Yeah, so it's a diagnostics company that takes samples, like kind of getting that back to the subway pole. When you have something that's in your mouth and you don't know what it is and you can't culture it, wouldn't it be great if you could sequence all the DNA or RNA and know exactly what's there? And so that company was founded on that very simple premise of, okay, people come in all the time to the hospital with infections that don't culture, it can't be diagnosed. If we break apart everything and sequence it, we can map it and figure out what it is and what it's resistant to. So we just got 2 different tests for the FDA last year, some related to COVID, not surprisingly. And also we have a new diagnostic test out in a lab that's here in Brooklyn, CLIA lab, for doing our diagnostics. So it's been a great company where the question is very simple. I want to diagnose what that infection is, but the methods had not yet been deployed on the market, nor put in a clinical lab. So we really got it out early, and now also a software package that can map viruses they move around the world. So it's been really fun to get that company going, and then we closed an A round just last year.
Yeah, congrats again. We're excited to continue on and see the exciting stuff that Biodia is going to keep doing, and especially in diagnostics. Need more of that all the time. So
yeah.
Taking a step back though, and just more looking at entrepreneurship as a whole, how do you think about crafting an entrepreneurial culture at your lab at Cornell specifically?
I've been really, you know, I feel like I've grown a lot in the past 10 years, because when I started the lab, I just want to have a lab and do my genetics and focus on the science. But the longer you have a lab, the more you realize some things can really only be done through a company in that mechanism. The best example is if you want to make 1 billion vaccines for, say, SARS-CoV-2, you by definition cannot do it in one lab or even at one university, right? It has to be through a large, well-capitalized company that can do it at scale. So I began to realize there's some questions that either need to be fast enough or large enough that are better served for companies, right? And so I think knowing that is just good for people to understand, for scientists and business and also just entrepreneurs anywhere. But then also telling it to students that there's not just one kind of job in their future. It could be, you could be a scientist anywhere. It could be at a nonprofit, a for-profit. It could be academic. It could be government. There's many, many places that you can do science, and you can do really cool and fast science within companies. And so I think getting them aware of that fact is part of the entrepreneurship, and then having avenues through which they can join a company, start a company, makes it possible. So Cornell has something called the Runway Program, Or when you get your PhD, or even if you get a master's, you just, you can put in and say, I've got an idea for a company. You get selected, you get 2 years of funding. That's actually how BIOS got started, was through this runway program at Cornell Tech, the engineering and tech campus right across the river here in New York City.
That's amazing. It's awesome to hear some of the genesis there, and really going beyond just founding your own companies and your own entrepreneurial lab culture. You're also an advisor to Tempest Labs. Can you tell us more about your work in just advisory roles, but also just the work specifically within Tempest?
Yes, I advise about 35 different companies, and Tempest is one of a larger, more chunk of time for the precision oncology and precision genetics. So it's a company that sequences cancer samples routinely and finds out what's causing and driving their cancer and treating it. So it is a really great offshoot of some work that's similar to here at our hospital. But again, they're at scale that is by definition impossible in most places, because it includes data from about 1/3 of all cancer centers around the United States. So you start to look at broader patterns of cancer, and you can do more machine learning and more complex analyses on those kinds of data. So it is a great company that also did some work in infectious disease for a while, but mostly oncology. And I was also doing some multi-omics work like we do with astronauts. So trying to get more and more of these molecular tools out into the clinic. And so it's a lot of fun to work with the teams there.
Yeah, it's amazing. Just as we come to a close here, I'm going to pass it off to my colleague Chris Bogdan to a few rapid fire questions to cap things off.
All right.
Thank you, Drew. And Chris, just 2 quick questions for you. First and foremost, do you have any other closing thoughts, shameless plugs? I know we've covered a lot of topics today, just given the breadth of your work, and it's been phenomenal having you. So I want to give you a chance to cover anything we might have missed.
I guess the shameless plugs. I think the book is the best synopsis of all the hopes and dreams that I've had at this point most of my life. So if you want to jump into my brain, also there's some really bad jokes in there about like bathtubs full of yogurt and odd things, but it's a pretty good synopsis of most of everything I think that we can do and should do as stewards of life in the universe. So I highly recommend it to jump in. And but yeah, I think you covered almost everything else. Every other book I've written or project I'm doing or company I've started or involved in. So it's pretty good, pretty thorough.
Oh, thank you, Chris. And how can our audience learn more about your work if they're interested in diving deeper?
Yeah, there's some work on the website. The book, of course, The Next 500 Years, Engineering Life to Reach New Worlds. Wherever books are sold, you can buy it. And also on Twitter, I guess at Mason Lab is at Mason_lab is either the papers I'm reading or thoughts I'm kind of thinking at conferences or tweeting out ideas from different scientific meetings is a good thread. Which who knows if the platform will survive given the recent mayhem, but hopefully it does, because actually it's a great place where people share methods and papers and ideas about science. And so it's a really fun community online there. So that's a nutshell. And then if anyone's out in New York City, I'm in the Upper East Side. Feel free to swing on by and we can swab you and sequence you.
Absolutely love that outro, Chris. And thank you so much for your time today. I'm sure our listeners had as much fun as we did, and we're going to be looking for you now at the next Poetry Slam. Thanks again.
Definitely. Absolutely. Thanks so much.
Thanks, Chris.